This is part 3 of Chapter 12's lecture, and now we're talking about anaphase. So anaphase begins suddenly when the cohesins that hold the sister chromatids together are cleaved or cut apart by an enzyme called separase. How's that for an appropriate name? There's the word for separase. Let me see if I can move this back in place. There we go. Once those chromatids are separate, we now have full-fledged chromosomes moving towards opposite poles of the cell. So there's been questions about how the kinetochore microtubules actually allow this movement of the chromosomes to occur. And what is believed that what's happening are one of two mechanisms. Both of these involve motor proteins. Recently, the scientist Gary Borisi of the University of Wisconsin suggested that the motor proteins on the kinetochores actually walk the chromosomes along the microtubules, which then depolymerize as the kinetochore ends after the motor protein passes by. Other researchers have shown that the chromosomes are reeled in by the motor proteins at the spindle poles, and that the microtubules then depolymerize after they pass by the motor proteins, or the motor proteins pass by the microtubule. It actually depolymerizes the microtubule. So that's how the spindle actually disappears. What is believed is that the contributions of these two different mechanisms or pathways actually varies among the cells according to the type of cell it is. But all of this is going to take ATP, which is really interesting. This is another diagram showing the kinetochores using motor proteins to walk the chromosome along the attached microtubule, and the microtubule shortening by dismantling the kinetochore. And I wanted to show you a quick just video of the idea of, just realized part of that got cut off, but what I was saying is I wanted to show you this quick little video showing a motor protein in case you haven't seen one before. So this is that really awesome video that was made showing the inner life of a cell, but this is the idea of a motor protein right here actually walking along the microtubules. And so it's thought in this case it's carrying a vesicle, but in the case of mitosis, they believe it's actually going to carry the chromosome, or yes, the now separated chromosome. Now this is actually going at this a third time, but I just want to make sure it's clear that when we have the double-stranded sister chromatids, that's one chromosome, but when they separate during anaphase, we end up with two chromosomes. So it goes from chromatids to chromosomes. So now we're done with anaphase, and we begin telophase. And the key characteristics here are the daughter nuclei are forming. So we have the reformation of the nucleus, and the actual chromosomes are dispersing. And really what that means is they're loosening up to not be that condensed form that we saw before. So the chromosomes have arrived at the opposite poles. The nucleoli are forming. We no longer can see the chromosomes under a light microscope because they are not as condensed as they were, and the spindle fibers have dispersed and disappeared. And almost immediately, cytokinesis is going to occur. In animals, cytokinesis happens by the constriction belt of actin, which are like microfilaments that go around the equator of the cell, creating a cleavage furrow. So animal cells, again, eukaryotic animal cells, are going to go through cytokinesis by making a cleavage furrow, which then splits the cell into two. And it's like tightening a drawstring around the cell and then pulling it tight until you end up cutting the cell in half. This division of the cytoplasm happens very quickly. On the cytoplasmic side of the cleavage furrow, that contractile ring of actin, our microfilaments that are very closely related to the motor protein myosin. So actin and myosin should sound familiar because they're proteins that are in our muscle fibers that are involved in contraction and relaxation of muscles. And so that's partly how this pinching of the two cells apart is occurring, is through a contraction. This picture is showing you actual photographs of the different steps of mitosis. So we have interphase here, and then we have prophase there. So the difference, notice the difference of the condensed chromosomes versus the loose ones. So we have the actual condensation of our chromosomes, that real tightening up that now makes them visible. Then in metaphase, we have them lining up along the metaphase plate. And in anaphase, they're starting to be pulled apart. In telophase, we have the beginning of the reformation of our nucleus. And then in late telophase, you actually do see the nucleus has formed, and then we have the beginning of the cleavage furrow. But before we go on, I have a joke for you. What is nitrate? It's lower than the day rate. Ha ha. And what is benign? It's what you be after you be ate. Ha ha ha. Okay, here's some more pictures. This one is showing prophase. In this case, you can see the nucleolus, you can see the nucleus, and you can see the chromatin condensing. So this is our nucleus here, that's the nucleolus. The chromosomes are out here and they're starting to condense. In prometaphase, now the chromosomes are quite visible, and we should have the beginnings of the spindle forming, but you can't see it very well in that picture. Here it's a lot more clear in metaphase. You can see the spindle fibers are getting those chromosomes, sister chromatids, lined up on the metaphase plate. Here you can see very clearly the spindle with anaphase pulling the two sets of chromosomes apart. Here we have a cell plate forming. So this is not a cleavage furrow. What is this type of cell? In telophase, in a plant cell, we have a different sort of cytokinesis occurring. So in plants, we have a cell plate that forms, and that forms through the deposit of vesicles that are laid down along the equator of this cell. So we've had anaphase just like we do in an animal cell. So we have the two sets of chromosomes separated out to the two different sides. But during telophase, what occurred were vesicles filled with cell wall materials, which came from most likely the endoplasmic reticulum, then packaged in the Golgi body. These vesicles move along the microtubules to the middle of the cell, where they actually join together to now form a cell plate. That new cell wall basically is being laid down by these materials between the membranes, and they're actually going to fuse into the existing cell wall until we have two separate cells. Here's a picture showing that process. So you can actually see the vesicles lined up in this photograph. It's a pretty awesome photo with the cytokinesis of a plant cell. So if you were to see a cell without even seeing the shapes, this rectangular shape tells you a lot about a plant cell. It gives you that and the big old vacuole over here. But another thing that would be a clue is seeing cytokinesis, seeing these little bubbles along the division plate right here, as opposed to a cleavage furrow if this was an animal cell. These are more pictures of plant cells going through different phases of mitosis. So you can quiz yourself and see how many you can figure out. but it's pretty clear on some of these what's going on. So mitosis in eukaryotes is thought to actually evolve from what occurs in bacteria or prokaryotes, which is binary fission. In bacteria, they have a single circular chromosome and they don't have membrane-bound organelles. So binary fission occurs in a very simple way, really. There still needs to be replication of the DNA with a prokaryote. It occurs, a chromosome replicates at the origin of replication on the circle of DNA, and then there's actually two circles of DNA that result, and the two daughter cells, daughter chromosomes, move apart from each other so that they can end up in the two daughter cells. There's probably a progression of mechanisms between binary fission and mitosis that we see in most modern eukaryotic cells. So there are different things along the way, different ways that mitosis occurs or cell division occurs along the way from simple bacteria, prokaryotes, to animal eukaryotes. For example, eukaryotes have spindle microtubules that begin to form between the centrioles outside the nucleus. And then as those centrioles move to the poles, the nuclear envelope breaks down and the kinetochore microtubules attach the kinetochore of the chromosomes to the spindle poles. And then the polar microtubules extend toward the center of the cell and overlap. In prokaryotes, at the other end of the spectrum, we have no nucleus, we have a single circular chromosome, and so after the DNA is replicated, it just gets partitioned in the cell. There's no spindle at all. After the cell actually elongates, there's a protein that assembles into a ring and helps separate and divide the cell in half. So in between these, we have some more unusual types of cell division. In dinoflagellates, diatoms, and some yeast cells, we may have some intermediary methods of cell division. Partly what happens in these types of cell division is that they do have a nuclear envelope, but it doesn't disintegrate during the division process. So again, in eukaryotes, we have the nuclear membrane disappear during prophase, but that doesn't occur with yeast or protists. The nucleus is present in a protist, which is also a dinoflagellate, and the nuclear envelope is going to stay intact during cell divisions. Protists have linear chromosomes like animals, and they have fibers called microtubules composed of tubulin protein. But the way that they actually separate out through this nuclear membrane, is that they pass through tunnels in the nuclear membrane and set up like an axis to separate the replicated chromosomes. The next type of intermediary organism is a protist. And so these are diatoms, and they get a spindle of microtubules that form between two pairs of centrioles at opposite ends of the cell. So now we're getting a little closer to a eukaryote. This spindle does pass through one tunnel because guess what they have? An intact nuclear envelope that does not disintegrate. So they have tunnels through their nuclear envelope for the spindle to pass through and facilitate the separation of the two cells in that way. Now yeasts are very simple eukaryotic cells, and they still have a nuclear envelope that remains intact. And they have spindle microtubules that form